Let's go ahead and get started. Thank you everyone for tuning in. I'm excited to introduce to you Trevor Carter, who's going to give the public portion of his defense for his master's thesis in botany. So Trevor received a double major in environmental science and forest management and ecology, and a double minor in rangeland ecology and ecohydrology from University of Nevada, Reno. That's more degrees than I ever got in my whole career. At UNR, he studied whitebark pine leaf traits in the Great Basin for his senior thesis. He came really highly recommended from his advisor, Beth Ledger at UNR. I remember she clearly said that if you need someone to climb a mountain, Trevor's your guy. And it turned out that my collaborator, Paula Fornwalt from RMRS in Fort Collins, and I were looking for just such an adventurous ecologist to take this project on. When I described the project to Trevor when he interviewed for the position, he was basically bouncing in the chair with excitement. And, you know, over the last few years, we've all come to know Trevor as a pretty incredibly energetic person who always looks on the bright side of things. He's also absolutely dedicated to finding the truth about plants and works really, really hard every day to improve his skills. And I think many of us would agree that if your field crew ever gets stranded in a mountain in a thunderstorm or a blizzard, he's the guy you want to be there in the group, because he'd be the one who'd be laughing and keeping our spirits high. And saying how funny it was, how that we would be so cold and uncomfortable. We're gonna miss having him around because he's off to University of Colorado, Denver to begin his PhD work this summer. But let's not get too far ahead of ourselves, because first he needs to pass his defense presentation. So without further ado, please welcome Trevor Carter.
Thank you so much, Daniel. All right, so disturbances are incredibly important for ecosystems. And this is especially true for forested ecosystems, where disturbance shapes the structure, function, and the identity of the ecosystem. And we can look at an extreme example from logging of a temperate rainforest near Vancouver in the city. So on the left, we have a skid road here near downtown Vancouver in the early 1900s. And on the right-hand side of the screen, we see present-day Thurlow Street, an approximate location for this left-hand photo. And so again, this is an extreme example of disturbance, and not all disturbances are that extreme. A lot of disturbances are not anthropogenic, but rather natural disturbances. And these are equally, if not more important for our ecosystems, because they still change the structure and function by maintaining the identity of this forest. And these forests are adapted to these disturbances. So we can see here a photo of a mountain pine beetle disturbance in Arizona. It's very easy to visualize this change in structure where we have these downed trees after the disturbance, the lack of a canopy where sunshine and light can come through into the forest floor. Other disturbances are incredibly important for maintaining the identity of a forest as a forest. And a prime example of this are the fires of 1988 in Yellowstone. And for those of you that were alive in 1988, they probably remember a lot of the headlines that were incredibly detrimental, that sort of painted this negative light of forest disturbance. But that's not entirely fair for the forest fires of Yellowstone. These forest fires were really important for maintaining the forest as a forest, like I said. Some research done by Monica Turner, a lot of research rather done by Monica Turner. She's sort of this rock star of forest disturbance, especially in the greater Yellowstone ecosystem, has shown these different recoveries of the forest after the 1988 fires. And these forests rely on these fires because they have these serotinous cones that basically melt in high heat to allow the seeds to disperse on the landscape. They also rely on the reduction of overstory to perpetuate their seedlings. So we see here just a variety of densities of lodgepole pines coming back after these 1988 fires. So now I'd like to just give a little bit of a talk outline so we can, keep me honest in terms of time, and we know sort of what we're going, where we're going and what we're doing. So I'd like to give a little bit of a background specifically for our forest. So our study took place in a high elevation forest, specifically the Glacier Lakes Ecosystem Experimental Site. And so I might refer to this as GLEAS throughout the remainder of the talk. And so this experimental forest is about 35 miles west of Laramie. For those of us not from Wyoming or the area, we see Wyoming here in the top yellow. We see Colorado here in the green, the national forest boundary in this dark green, and the approximate location of the Glacier Lakes ecosystem. And so this Glacier Lakes ecosystem is a 600-hectare experimental study area with the US Forest Service. And like I said, it's high elevation. Specifically, it's a subalpine spruce fir forest. This is a very common forest type, not only in the Southern Rocky Mountains where we are, but also the Northern Rocky Mountains, parts of the Cascades, a lot of Canada and interior Alaska. When we think of these subalpine spruce fir forests, there are two major disturbances that come to mind: fire and spruce beetles. And these spruce beetles are similar to mountain pine beetles, but I'm going to go into depth on those in a second. And fire and spruce beetles have some similarities. Both cause wide-scale mortality across the landscape, and both respond to similar climatic triggers. Because these disturbances respond to similar climatic triggers that are relatively rare, we generally think of these disturbances as having a low frequency. And there's a trade-off between frequency and magnitude of disturbance, or severity rather. So when we have these low frequency disturbances, they tend to be very high magnitude or very severe, affecting a lot of the landscape. But when we have these relatively high frequency disturbances, they tend to be relatively low in magnitude. And like I said, both fires and spruce beetles have these similar climatic triggers that make them infrequent and severe. And so I've been talking about these climatic triggers, but what exactly are they? And looking at this picture, it's pretty easy to recognize that we are in a cold and wet system. This photo was not actually taken in winter. This was taken June 1st, a couple of days after I moved to Laramie, looking over my study site. So we have here Medicine Bow sort of behind these mountains right there. And in this corner of the screen, we have the Glacier Lakes Ecosystem Experimental Site. And so we can imagine June 1st, it's very cold, it's very wet. And if we think about these cold and wet climates, we can imagine that it's very difficult to light a fire when it's really cold and wet, and a lot easier when it's warm and dry. These climatic triggers are very similar for spruce beetles, but a bit more complicated because they are a biotic agent of disturbance. So spruce beetles in the Rocky Mountains are the most significant disturbance for mature spruce. And when I say that, it's spruce beetles in association with their fungal associates. And they have a very complex ecology, and I could probably spend a whole 50-minute talk just talking about how interesting and fascinating these spruce beetle the spruce beetle ecology is. But to break it down for relatively short purposes, we basically have this really interesting complex life cycle where we have these mature adults that need to overwinter before they lay eggs inside the bark of spruce trees, mature spruce trees. These eggs then hatch as larvae. They overwinter again before they pupate and turn into immature adults. And so this is a 2-year life cycle. But when we have less harsh winters, or winters that are warmer and drier, this life cycle can condense into a single year. And we can see these changes in the population dynamics from going from these endemic populations that are only really capable of causing mortality in sort of old and really big trees, to these epidemic population explosions that overwhelm tree defenses and cause landscape scale mortality for, again, our mature spruce. And from this range map, we can see that this is not isolated to the Southern Rocky Mountains. This is, again, through the Northern Rocky Mountains, a lot of Canada, and interior Alaska. And as climate changes, we can only predict that these epidemic spruce beetle outbreaks are going to get worse. So why do we even care about epidemic spruce beetle outbreaks? And that's a great question. So we have here a picture of our forest. This is before the disturbance. And if we saw a fire across this landscape, we might imagine a complete loss in overstory structure, a change in the understory community that would get affected by the disturbance as well. Our soils would be affected. But because we saw spruce beetles, and spruce beetles have this proclivity or this preference for these large diameter spruce trees, we see something quite different. And this is a bit of a dramatic change. And so I want to make sure we can all visualize this, that this is a large change in our ecosystem, but only by the reduction of our mature spruce trees. And when we have the loss of our dominant competitor on the landscape, a lot of interesting observations and questions can then arise. And one of these interesting observations came from the Carter perspective. And so this is a bit of a complicated figure, so we're going to walk through it slowly. And we really only need to be focusing on this green section, or our net ecosystem exchange. So we have here net ecosystem exchange, or basically our carbon through time. So carbon fluxes, where we have this zero line. Positive values would indicate carbon leaving the landscape or through respiration, whereas our negative values are carbon being sequestered or fixed by the landscape. And these are, our photosynthesis. And so in 2005, before the disturbance, in our winter months, we see that our carbon fluxes are relatively close to zero, that there's not a lot going on. Our plants are dormant. Remember the picture that I showed just a couple seconds ago? It's pretty cold, it's pretty wet. But once it becomes summer, we see these large draws of carbon into our landscape. So plants are waking up and they're photosynthesizing a lot. And so this was, again, before the disturbance. And after the disturbance, we might anticipate something very similar to this winter month section, since our dominant competitor has been removed from the landscape. However, that's not exactly what we see. In red here, we now see our net ecosystem exchange after the disturbance. And these are relatively similar figures, where we see in our winter months that not much is going on. Our plants are pretty dormant, but then again in our spring, late spring, early summer, our plants start to wake up and a lot of photosynthesis is happening. And this is really interesting because again, our dominant competitor has been removed from the landscape. And that brings us into our interest in the understory plant communities. And understory plant communities have been shown to be important for these carbon fluxes. Specifically, some research done by Mission et al. 2007 saw that these understory plant communities were significant contributors to nighttime CO2 fluxes. And so let's break down this figure as well. So on the left side of this figure, we have our evergreen forests, which is our conifer forests or our spruce fir forests. So we really need to focus only on this section here. We have our nighttime CO2 fluxes both above the canopy in black and below the canopy in dark gray. And we can see that our below canopy represents some significant portions of the above canopy nighttime CO2 fluxes. Mission et al. Broke it down a little bit further and looked at the specific percent contribution of understory plant communities in these evergreen forests. And we see that understory plant communities contribute at least 38% of these nighttime CO2 fluxes, but upwards of 60%. And so these understory plant communities are also really important for our forest regeneration. Some research done by Petit et al. Saw that regeneration was potentially driven or positively associated with understory plant cover. And this is really important because the regeneration of our forest is particularly uncertain. As climate changes, we might not be able to regenerate our dominant tree species. And so something that I would like to point out in this slide, that despite the size in this picture, seedlings of this size likely predate our disturbance. These seedlings might be even as old as I am, even though they are only a handful of centimeters tall. And so we measured these size classes because going less than this size would be really difficult to create accurate measurements for, and over this size might not necessarily be representative of vulnerable seedling sizes. And so I just want to break this down a little bit further. On the left hand, we see this table that shows understory cover as being a significant predictor of our advanced regeneration. Things here that were predated from the disturbance, where essentially where there was more understory cover, there was more advanced regeneration. And this was research done by, again, Petit et al. On the right hand, we see this recruitment of year one for our Engelmann spruce, which is our dominant spruce trees. And this is some research done by Coopers et al. 2017. We really only want to focus on this panel A, specifically on this first set of bar graphs in our forest environments, where they added heat treatments to their plots. They saw significant reductions in the recruitment of year one seedlings for our Engelmann spruce. Again, these are our dominant trees on the landscape, and so it makes us question whether or not these forests will return to their pre-disturbance states. Lastly, understory plant communities are really important for our wildlife communities. And while I don't have any research that actually links understory plants to moose, this is sort of a solicitation for any papers related to understory plant communities and moose populations. I do have a lot of other research related to specific pollinator communities. And so some research done by Davis et al. 2020 essentially saw that when we have these reductions in canopies and more open canopies, we see increases in our floral abundances, and this is associated with increases in alpha richness and diversities for our bee communities, specifically a 54% higher bee abundance and a 44% higher alpha diversity in locations with reduced canopies. So I'd like to talk a little bit about what we did for our specific study. So we collected data with these three questions in mind. First, how has understory plant cover and richness changed since the disturbance and across a gradient of spruce mortality? Second, have spruce and fir seedling densities changed since the disturbance or across a gradient of understory cover? And third, what mechanisms predict the response of understory plant cover? And so to get at these questions, we resampled these 75 plots throughout the Glacier Lakes ecosystem. And these plots were originally sampled in 2010 by our co-authors, Dr. Paula Fornwalt, Dr. Kate Dwyer, and I am incredibly appreciative of their sampling efforts and being able to use this data. We also got incredibly lucky that not only did these plots represent a gradient of elevation and aspect, but these plots represented a naturally occurring gradient of spruce mortality. Because this is a botany seminar and I'm hopefully getting my master's in botany, I thought it would be appropriate to talk about some of the plants that we saw on the landscape. And so by far the most dominant plant before the disturbance was this Vaccinium scoparium, or grouse whortleberry. Some of us might be familiar with it. It's this sort of cespitose sub-shrub that occurs across the landscape, creating these large mats. It's in the blueberry family, so it's got these deliciously edible berries in about late August that you can find across the landscape. Other big contenders were our Rigeron peregrinus, or subalpine fleabane, our Arnica cordifolia, or heart-leafed arnica, and our Sebaldia procumbens, or creeping Sebaldia. So to actually collect these data, what we did at all 75 of our plots is establish these three transects that were established on the same azimuths as they were in 2010. And I actually really like this video. It showed us that even after dozens of plots in the field season that we were still prone to make mistakes and set up the azimuths on the wrong bearings. So we corrected that, but such is field work. So to collect data on our understory cover, we had 5 evenly spaced Daubenmeier quadrats for each transect. And so these Daubenmeier quadrats are these 20 by 50 centimeter frames that we can use to estimate percent cover. You just saw my field technician Mason walking around with a seedling pole to calculate, or count rather, how many seedlings were within our plots. We also counted richness, we estimated canopy cover, and we collected diameter at breast height for all of our trees within the plot. And so this was a huge sampling effort, and I would like to take a little bit of time within the talk to just say thank you to not only my field technicians, Mason and Carl, but also the lab for helping out. The restrictions, we were able to get really lucky that we were able to abide by COVID restrictions and still collect 75 plots worth of data in a single field season. And so to recap, the data that we did collect was canopy cover, plot richness, understory cover, and our diameters at breast height. And something that we did with this diameter at breast height, and something that I'd like to make note for those of us that might not be foresters or forest ecologists, our diameter at breast height is this standardized point on a tree, 137 centimeters, that we measured the diameter. And we use that diameter to estimate our basal area of our tree. And this is an estimate of sort of densities and sort of the competitive influence that trees might have on the landscape, because we can use this through geometry, estimating our diameters into our basal areas. And so now I'd like to talk about what we saw. So our first question, how has understory plant cover and richness changed since the disturbance and across a gradient of spruce mortality? So first, we're going to look at how understory plant cover has changed since the disturbance. We see here on the left our total plot cover, both before the disturbance in gold and after the disturbance in blue. We see our plots within each of these bars. And so we can see that this was actually a statistically significant increase after the disturbance. And this increase was about 16% increase in cover on average. We also ran some linear models to estimate or to sort of uncover what some of these significant predictors of this increase were. We saw that basal area was a significant predictor. Where there was less live basal area, there was more associated increases in plant cover. And so where there was more live basal area, there was less increases in plant cover. And this increase is actually really evident when we look at one of our most extreme plots, our plot GL0374 that experienced 100% mortality. We see the same transect in the same plot before the disturbance and 10 years after the disturbance, Again, same plot, same transect. And I think it's really fascinating to see these increases in cover and also the breakdown of these logs. And when we look at the increases by a species and functional group perspective, we see that our Vaccinium scoparium, or that subshrub that I showed earlier, it was really dominant before the disturbance, and again, really dominant after the disturbance. And another shrub, our Ribes montigenum, that was sort of dominant before the disturbance, was again really dominant after the disturbance. And that fed into this importance for shrubs being the most dominant functional group before the disturbance and most dominant functional group after the disturbance. And this increase was large, but only about a 35% increase. When we looked at the increases in our forbs, we saw about a doubling of forb cover on average. And then when we look at our graminoid cover, we see about a 5-fold increase in graminoid cover. We can now look at how understory richness has changed since the disturbance. And so again, we see our number of species, both before the disturbance in gold and after the disturbance in blue. And this one is a little bit easier to visualize that this was again a statistically significant increase. In fact, understory richness more than doubled following the disturbance. And this equated to an increase of about 10 species on average per plot. Again, in our linear models, some of our significant predictors were elevation and aspect, such that lower elevations in more southerly facing aspects had more species than high elevations in northern aspects. So we can also look at how understory plant cover has changed across a gradient of spruce mortality. And I think that this is really fascinating and something that comes sort of as a real power of our data set, since we were able to resample the same plot both before and after the disturbance. So on this figure, I've highlighted this dashed red line, which is the 0 line. Anything to the left of this are reductions in live basal area, or mortality within our plots. Anything to the right of this are increases in live basal area, or growth into our plots. And so we see our change in understory cover as a function of our change in live basal area. Where there was larger reductions in our live basal area, there were larger increases in understory cover. We also evaluated how understory richness has changed across a gradient of spruce mortality. And so again, we see a similar trend where we see larger increases in species richness associated with larger changes or larger reductions in live basal area. However, this was not statistically significant. What was statistically significant for this specific model was again elevation and aspect. Lastly, we were able to look at how both understory cover and richness changed across a gradient of spruce mortality. We did this by using a plot dissimilarity metric. And this is sort of a multivariate data metric that incorporates both cover and richness. And so to break that down a little bit, imagine if we had one plot that had low cover and richness before the disturbance and low cover and richness after the disturbance. That plot would be very similar to itself and have a low dissimilarity score. However, if we had a plot with low cover and richness before the disturbance and high cover and richness after the disturbance, that plot would be very dissimilar to itself. And we see essentially that plots that are more dissimilar to themselves were predicted by larger reductions in our live basal area. So to summarize, how has understory plant cover and richness changed since the disturbance and across a gradient of spruce mortality? We saw increases in both cover and richness. Understory cover increased across a gradient of spruce mortality, and richness was not predicted by spruce mortality. So our second question, have spruce and fir seedling densities changed since the disturbance or across a gradient of understory cover? Again, this understory cover comes from our literature. So we can look at our spruce seedling densities and how those have changed since the disturbance. And so we see our spruce seedling counts both before the disturbance in gold and after the disturbance in blue. And these changes aren't that big. And this is actually a non-significant difference. So spruce seedling densities did not change following the disturbance. And this is sort of a relief, right? If we're worried about the future regeneration of our forest, they were low before the disturbance, but they were still low after the disturbance. At least they didn't significantly decline. We can look at our fir seedling densities and how those have changed since the disturbance. And so again, we have our fir seedling counts both before the disturbance in gold and after the disturbance in blue. And we see a lot more variation before the disturbance. This change was statistically significant, and there was a decline following the disturbance. On average, there was a decrease in about 5 seedlings. We can again look at how our spruce seedling densities have changed across a gradient of understory cover. I've highlighted our 0 lines, so these are no changes in our seedling densities. Anything above this 0 line are increases in our seedling densities, and below this are decreases in our seedling densities, as predicted by our change in understory cover. And for our spruce, we see a lot of these densities hovering around 0, not that much change, and understory cover not being a very good predictor of this. We can do this again for our fir seedling densities. And we see that this is a very similar thing, where a lot of our seedling densities are hovering around this 0 line, but a loose trend of increases in understory cover leading to less losses in our fir seedling densities. But understory cover in general is not a great predictor. So to summarize, have spruce and fir seedling densities changed since the disturbance or across a gradient of understory cover? We saw no change in our spruce densities. We did see a decline in our fir densities, and understory cover was not a great predictor of seedling densities in general. So now on to our third and final question. What mechanisms predict the response of understory plant cover? And we evaluated 2 potential mechanisms. Our first mechanism was priority effects. And so to sort of break down what these priority effects are, we can imagine that we have a plant community composed of species A, B, C, and D in 2010, where species A was the most frequent and species D was the least frequent. 10 years after the disturbance in 2020, because species A was the most frequent, it has the largest change in cover, whereas species D has the lowest change in cover. And so this is a neutral model where species change in cover is not driven by a species identity, but its inertia. So the other mechanisms that we evaluated were plant functional traits. And if you know the Lachlan Lab, this comes at no surprise that we were looking at plant functional traits. And so essentially a plant functional trait is any trait that may confer a fitness benefit for this plant. So these functional traits that we evaluated, the first one that we'll talk about is plant height. And I like this picture because it sort of shows a variety of understory plant heights within this photo. So in the background, we can see our taller understory plants. And in the foreground, we can sort of see these lower or more cespitose understory plants. And so understory plants that are taller might be having higher quality light or access to more high quality light than plants in the foreground that are really small and sort of have access to this diffuse light that has to pass through these taller understory plants. It's very similar to playing basketball, where if you're really tall, you're really good at keeping the basketball way up high. And if you're really short, you're probably not going to be having a good time playing basketball. Another set of functional traits that we looked at, this one was specific leaf area. And this is derived from the leaf economic spectrum, which is essentially a continuum of values from more acquisitive plants to more conservative plants. And so specific leaf area is the measurement of the leaf area per mass. And what's really important to remember is that this is a metric per mass and across a continuum. And so I have these hypothetical values for these 2 hypothetical plants to sort of demonstrate these values. So we might have a more acquisitive plant with these really leafy leaves that's able to photosynthesize a lot, that would have this really high specific leaf area value, as compared to this more conservative plant that is sort of these less area per same unit mass, and that's maybe more conservative with its resources. And these generally, I think of them as trade-offs between being more acquisitive or more conservative. Another functional trait that we evaluated was leaf turgor loss point. And a leaf turgor loss point is the water potential that plants lose function in their leaves. And you might be asking why we are looking at a drought tolerant trait if it is a really cold and wet environment and snow is possible at any month of the year. And that's a really good question. Although it is really cold and wet and precipitation is indeed possible in any month of the year, these soils are really shallow and poorly developed. And so they still dry out at the end of this very short growing season. And if plants are able to sort of withstand a little bit more or withstand lower turgor loss points, and able to persist longer in the growing season, that might confer a fitness benefit over neighbors. Additionally, we might anticipate our vapor pressure deficits to increase as a function of this disturbance. So some research done by Reed et al. Saw that with increasing mortality in a nearby lodgepole pine stand, that vapor pressure deficit or this atmospheric demand of water increased. And so having more drought tolerant plants might be able to survive these conditions and thrive a little bit better. Lastly, we evaluated dispersal mechanism. And we did this because dispersal is often a key constraint to plant community assembly and widespread dispersal may aid a species in increasing in cover. So to summarize our functional traits, we looked at height, we looked at specific leaf area, we looked at leaf dry matter content, which is another trait associated with the leaf economic spectrum. I'm not going to talk about this too much. We looked at leaf turgor loss point, and we looked at dispersal mechanisms. And we did this because functional traits represent, especially these functional traits, represent a variety of ways that plants can be responding to resources, either light or water. And these are also commonly used traits with a lot of literature to support them. So specifically, when we looked at our question, we asked, do priority effects, functional traits, or both explain a species' response to beetle attack? We did this because these two mechanisms are likely not mutually exclusive, and they both could be playing a role. And that's actually what we saw. And so this is, again, a big figure, so we're going to walk through it. On each side, we see our change in species cover as a function of our specific mechanism. So in panel C here, we have our pre-disturbance species frequency, or analogous to our priority effects. Something to note, I have highlighted the 3 most dominant species after the disturbance in each of these plots, just so we can sort of visualize where they are on this space. So for our first plot, we see our change in species cover again as our pre-disturbance frequency. So if you were more frequent across the landscape before the disturbance, you increased in cover the most after the disturbance. We also saw that taller plants, or plants with taller heights at maturity, had larger increases in cover after the disturbance. We saw that our species with our lower turgor loss points, or our more drought-tolerant species, increased the most in cover after the disturbance. We also saw this intermediate value for our specific leaf area, where species with these intermediate specific leaf areas increased the most, but on either end, there wasn't these large significant increases. So to summarize, within our functional traits, we saw that height, specific leaf area, and leaf turgor loss point were our significant predictors, and our leaf dry matter content and our dispersal mechanisms were not. So to summarize this, do priority effects, functional traits, or both explain species responses to beetle attack? Both do. And in terms of our traits, we saw height, turgor loss point, and specific leaf area as being important predictors. And so now I'd like to give a quick recap and sort of do a little bit of supposing about what some of these results might mean. So how has understory plant cover and richness changed since the disturbance and across a gradient of spruce mortality? We saw increases in both cover and richness, and understory cover increased across a gradient of spruce mortality, and richness was not predicted by spruce mortality. And this was sort of anticipated based on the literature of other disturbances, where basal area is a good predictor of our increases in understory cover, specifically after fires and mountain pine beetle outbreaks. Something that might be important for these increases in cover, specifically our increases in graminoid cover, could be for our wildlife communities, specifically elk. Some research done by Lamont et al. 2019 saw no changes in elk movements or increases in their behavior towards graminoid dense areas because of increased movement costs associated with downed trees. However, we don't really have that many downed trees 10 years after the disturbance, so this might be changing how elk move across the landscape. Our second question, have spruce and fir seedling densities changed since the disturbance or across a gradient of understory cover? We saw no change in spruce densities, and we did see a decline in our fir seedling densities. We saw that understory cover was not a great predictor of seedling densities. This third bit is really fascinating to me because it directly disagrees with the literature, which is always fun because that means we can study this more. Our timescales for disturbance were 10 years after the disturbance, and the research done by Petit et al. Was 20 years after the disturbance. So maybe our timescale is important and that we're operating on a smaller timescale after the disturbance. And in this sense, that understory cover is not a great predictor of seedling densities in this timescale. And it's interesting to suppose why. Our third question, do priority effects, functional traits, or both explain species response to beetle attack? And we saw both. Specifically in terms of our traits, we saw our height, our turgor loss point, and our specific leaf area. And something that I would love to understand a little bit more is why our dispersal syndrome was not an important predictor. And this might be because some increases in cover didn't actually result from new individuals. Maybe our Vaccinium scoparium, this cespitous shrub, didn't reproduce a lot and was just mostly growth. And that was potentially why dispersal mechanism was not important. For other plants, maybe like our Rigeron and our graminoids, dispersal is incredibly important. So that is yet to be teased out. So lastly, I'd like to put my research into sort of a bit of a broader context of the field. Something that, again, is really interesting with this research is its similarity to other findings. Specifically, this research done by Joel McMillan in 2001, looking at the effects of Western balsam bark beetle, which is a very similar bark beetle, on spruce fir forests in north central Wyoming. And so Western balsam bark beetles attack fir, which is, again, not our dominant species. But in this research, they saw that understory graminoid cover had a 5-fold increase, much like ours did, and understory forb cover had a 2-fold increase, much like ours did. And this is incredibly interesting to me because disturbance and the results of disturbance studies are generally not generalizable. There aren't a lot of very generalizable findings that can be applied to a lot of these different disturbance studies very easily. And so having a near match for our increases in forb and graminoid cover, I think is really interesting and maybe provides evidence that functionally understory plant communities are responding very similar to these two disturbances. I talked a little bit about the interesting findings that we had with understory cover not being a significant predictor of our seedling densities. To sort of tease this apart a little bit more, I will be working with a research group out of Madison, Wisconsin, specifically Dr. Carl Rodman, to sort of look at these large drivers of seedling densities and seedling retention after these spruce beetle disturbances in the Southern Rockies. And so this is going to be a really fun, big project that I'm excited to see where this one goes as well. And lastly, something that I think is really sort of a hot topic issue is this interaction between spruce beetles and fire. And this is not a new topic of debate. This has sort of been going on for the last 20 years with very mixed results. But understanding how understory plant cover has changed since the disturbance provides potential new information for how fires might be behaving or changing after spruce beetle disturbances, specifically because they increase the continuity of fine fuels for longer timescales after the disturbance. However, there's still a lot of research that needs to be done into this. And so who knows what we'll find if we evaluate the effects of understory cover in these models. And so I would like to say thank you to a few people in particular, specifically my advisor, Dr. Daniel Laughlin, my committee, Dr. Brent Ewers and Dr. Kevin Wilcox, the Laughlin Lab, my field technicians, Mason Parkus and Carl Lenore. And with the U.S. Forest Service, my co-authors, Dr. Paula Fornwelt and Dr. Kate Dwyer. And I would especially like to thank Dr. John Frank for a lot of help in thinking through parts of this project and pictures used in this presentation. And so with that, I would really like to say thank you all for coming. I am very appreciative and I would be happy to take questions.
All right, thank you very much, Trevor. We will open it up for questions now. We have plenty of time. You can either unmute your mic and just go ahead and ask your question directly to Trevor. If you feel more comfortable, please feel free to throw the question into the chat and I will be monitoring that.
I have a question in the chat from George Jones. So in figure 1, changes in understory cover versus change in understory live tree basal area, and 2, change in understory species richness versus overstory live basal area. It looked as though some plots had increases in both understory cover and understory richness, where overstory tree live basal area increased. What's going on there? That is a great question. Let's see if I can pull up the figures so we can all visualize that. I want to also make sure we're on the right one. So I think this is the figure that we are discussing. This figure and this figure as well. And there are these increases in species richness despite there being increases in live basal area. That's a really great question. To me, this says that the effects are across the landscape and not isolated to these plots. And so although these plots are about 0.1 hectares, they're not isolated necessarily, where we might have seen increases in live basal area directly within the plot. Just outside of the plot, there could be reductions in live basal area, and those effects could be influencing inside the plot. No plot is an island, and the effects of the landscape are going to influence this across all of the plots.
Trevor, I have a question for you.
Hi, Alicia. Yeah.
Great presentation, by the way.
Thank you.
So you touched on this, but it seems like the timescale at which you did your post-disturbance sampling really influences perhaps some of the things that you'll find. So you mentioned like seedling densities. But besides that, are there any other conclusions that you came to that might be affected by the timescale you chose to do your post-disturbance sampling? And could you talk a little bit about that?
I'm not entirely sure if I understand the question, because I think the timescale for our disturbance and our responses are adequate because our understory plant communities have had the chance to respond to these changes in live basal area. The canopy, something that's really important for our spruce beetle disturbances is this drop of the canopy that goes from green phase to red phase, where spruce beetles have attacked the trees and they are in the process of dying, and then into this gray phase where they are dead. And so I think that our timescale is appropriate to evaluate all of these questions. But I think that as we move forward in time, these processes might change. As this ecosystem develops after the disturbance, we might see different results moving forward. Does that sort of answer your question?
Yeah, thank you.
Hey, Trevor, great talk. I had a question about some of the trait results. And so in my work, we found a pretty tight correlation between leaf dry matter content and turgor loss point in like drier grassland and shrubland systems. But that did not seem to be the case in this system. So do you have any hypotheses that might explain that?
Yeah, that's a great question. My knee-jerk reaction is to think that our turgor loss points aren't quite as, they don't quite have the range that you might see in drier systems, since this is not really a particularly water-limited system. And that I think our range between our turgor loss point is just 1 megapascal of turgor loss point. So it's not a lot of range within that, and I think that that might be influencing these correlations between different traits. I have a question in the chat from Jared Merkel. My question is, which of your metrics, if any, vary by year, and what are the implications of that for your conclusions? That's a great question. So I think year to year that our cover measurements would not vary greatly, or our richness measurements, since these systems are generally categorized by a slow response to disturbance. But when we look at multiple years, like in the 10-year plus time scale, that's where we see these changes occur. So I think that if we were to go out and sample these understory cover and richnesses this year, that we would see very similar, if not the same, probably not the same, but very similar results across the landscape.
Hey, Trevor. Nice job, man. I got a question for you. So obviously between comparing priority effects and trait measurements, one is a little more laborious than the other to gather. Could you give an example of maybe where you need the traits to, like a project or a question where you need traits to answer? Is that more of a conservation thing? Or if like when you're concerned primarily with the net ecosystem exchange, are cover estimates enough to make an estimate of how the ecosystems are going to respond?
Those are great questions. Thank you, Dave. So from my limited understanding of how net ecosystem exchange has been evaluated in some of these systems, specifically with influence of understory cover, a proxy for understory leaf area has been used to sort of evaluate these changes in, or the importance of understory plant communities for these carbon fluxes and net ecosystem exchange. So I would think that understory cover provides a decent enough proxy for these changes in carbon fluxes. And then in terms of your first question, in terms of what use some of these traits might be for conservation, I think that traits can provide us a more detailed information about what nutrients or what resources plants are using and responding to. specifically because we saw Turgor loss points and lower Turgor loss points being significant predictors. That might indicate changes in snowpacks and vapor pressure deficits to further evaluate and sort of see the vulnerability of these ecosystems to those processes that influences those resources. Hopefully that answers your question.
Hi, Trevor. Great presentation. I have a question for you. So you've been working with this management agency, much like I do, the Forest Service. So I'm curious what you think some of the management side implications are for this work. I think it seems like there's probably a lot to be said there about the seedlings not responding to the understory, but maybe there are some other insights you've had for management as well.
Yeah, that's a great question as well. I think in terms of management implications, I like to think of a more hands-off management approach. If we don't have to do anything to this system, if we don't have to invest in managing the system, if the system seems like it's responding in a way that would lead to pre-disturbance conditions, then we shouldn't do anything. And I think that this provides some evidence that the system at least isn't being detrimentally affected. Like there was no change in our dominant spruce seedling densities. And so to me, this doesn't ring any alarm bells that we need to be out there planting spruce seedlings. Although I think that some further research needs to be investigating the specific drivers for seedlings. For us, seedlings were not necessarily the main focus. Understory cover was definitely the predominant focus. And I think we learned some insights about these seedlings and how those have changed. But I think in the future, for very conservation and management applied research, we would want to evaluate the effects of seedlings and look at them a little bit more intensely.
All right. Any further questions for Trevor? All right. Well, seeing none, we will adjourn to the closed portion of the defense, but please join me in thanking Trevor and for his wonderful presentation. And thank you all for coming.